Okay, guys, we're back, picking up where we left off. This video is going to be talking about how the neurons communicate with each other, what happens at the synapse, and we'll probably get into some of the different neurotransmitters. If you're not sure what I'm talking about, or if this image looks unfamiliar to you, then you need to go back and watch the previous video about the structure of the neuron and kind of an overview of the nervous system. So again, this video will be about the function of the neuron so that we can understand the role that it plays in our nervous system and how it communicates to other cells. So first off, neurons generate what we call a neural impulse, or to use more technical terms, an action potential that sends an electrical charge down the axon to the terminal ends that then communicate information to the other neurons around them. So first off, this is a chemical process. So you have positively and negatively charged sodium, mostly sodium ions, sometimes potassium ions as well. And you can think of a neuron as being both made up of internally, like the stuff inside of a neuron, and swimming in what we call cerebrospinal fluid. It is basically this fluid that makes up both the outside and the inside of the neuron. So at rest, what we call at rest, there is positive neurons on the outside of the neuron, positive ions on the outside of the neuron, and negative ions on the inside of the neuron. And as if a neuron, which we'll get to in a second, decides to, for lack of a better word, fire, right, to release its neural impulse, to create an action potential, to depolarize, all terms are synonyms here, it actually brings in the positive ions from outside of the axon, and it then replaces it with the negative. And you can see in this image here that it travels down the axon, right? Neural impulses only go one way. This is important. Axons are not a two-way street. They're a one-way street. So information can only go from the soma to the terminal ends. It can never go the other way around. Okay, the axon only works in one direction. So as these ion channels open up and change the polarization, right? They become depolarized, right? Polarized meaning you've got positive on one side, negative on the other. Once you see that mix, that's called depolarization. And you can see that there's this kind of give and take of positively and negatively charged ions. First, the positive ones come in, then they're kicked out by the negative ions, and that forces the neural impulse down the axon to the terminal ends. Now, all of this takes place in not even milliseconds, faster than that, right? It's one of the fastest things in nature, the release of information within a neuron, and it's all a chemical process. So a couple other things we need to talk about with the action potential, some terms that apply to all neuron firings, okay? First is something that we call the all or none law. So what this means is that neurons, when they fire everything they got. They don't leave anything back. There's no such thing as a weaker impulse, okay? When a neuron fires, it releases it releases the electrical charge completely. Okay, it does not kind of do it weakly or it's a half fire or whatever. It's all or none. It either fires or it doesn't. And what's happening is, remember, neurons don't exist in isolation. They're surrounded by most of them are surrounded by other neurons, or they could be surrounded by they could be sensory neurons that are taking information in from our eyes, ears, skin, etc. So basically what the cell body is doing, again, think of the dendrites that are connected to all these other neurons. Those other neurons are communicating to, say, our main neuron that we're focusing on here. It's basically tallying up all of that information, right? So some neurons are saying, hey, fire, send this information along. And other neurons are saying, don't, stop. So what's happening in the soma essentially is it's keeping track of the should it fire or should it not, right? And at a certain point, it's going to hit a threshold, and that threshold is going to require it to fire. If it hits that threshold, it fires every time. It's automatic process. Okay, um, so we have a term here called a threshold, that once the neuron receives enough information to fire, right? Once it passes that threshold, it will fire every time. And again, it is all or none. So it's the same strength down the axon. It doesn't matter if it is told to fire like by every neuron that's connected to it or by half of the neurons, but once it hits that threshold, it fires. Okay, um, and again, it's going in the one direction down the axon. Afterwards, after a neuron fires, there's what we call a refractory period. That refractory period is that the neuron basically needs a little bit of time to recharge. It can't fire again, no matter what the other neurons are saying to it. So if it's receiving all this information from other neurons like fire, 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 it's not going to if it's still in the refractory period. It essentially needs time to recharge as the ions get replenished and it we call repolarizes, right? It goes back to a polarized state. Okay. So one of the things, this is going to be a homework assignment. One of the things that I want you guys to do is to think about the relationship between neurons and how neurons work and how neurons operate and how a toilet operates. So there's going to be an assignment on Schoology where you're going to have to write some of these terms that we've talked about, both in our book and in these notes, relating to neurons, and you're going to have to apply that to the way a toilet works. So your homework literally is to go to the bathroom. And we'll talk more about that in class, but just telling you now that that is one of your homework assignments for this unit. Okay, so let's talk about what happens once the action potential leaves the cell body, travels down the axon, and is ready to interact with another neuron. So again, we're back to the same images we were using before. Okay, so the neural impulse gets translated from a neuron to the other cells. That transmission takes place at the synapse, which is where, again, the terminal buttons or terminal ends or whatever you want to call it, release chemical messengers that we call neurotransmitters. Okay, there is a microscopic synaptic cleft that, if we zoom in, look at that animation. Wow, where the neurons are actually not touching. Okay, so the dendrite kind of comes in, as you can see here, and the synapse is kind of, or the dendrite has kind of got like an indentation here. So it's not actually touching. It is a microscopic gap in between the two. And that's really important because this is crucial to how information gets spread from one neuron to another. Okay? The signals, the chemical signals have to cross that synaptic gap in order for the information to be received. Okay? So let's get some terms down now. So the neuron that is firing, the neuron that is sending the information is called the presynaptic neuron, right? It's before the synapse. It is the neuron before the synapse. The postsynaptic neuron is the one that's receiving the information, right? It is after the synapse. So if you think of the synapse as the main intermediary here, there's the one that's before, and there's one that's after. Okay, more terms. There's something called the synaptic vesicles, okay, which is a fun word to say. Synaptic vesicles. A synaptic vesicle is a small sac of neurotransmitters. So the neurotransmitters are, again, chemical messengers. Neurotransmitters are the chemical messengers that the neurons are sharing with, the presynaptic neuron is sharing with the postsynaptic neuron. They are stored in synaptic vesicles, okay? And those are found within the terminal ends, terminal buttons. So they just kind of, the terminal end is kind of filled with these, sacs of chemical messengers that then basically spew out these neurotransmitters when necessary. Okay. So what happens at the synapse? Let's take a little animation here. So in comes the action potential, right, to the terminal end. It says release the neurotransmitters, the chemical messengers, right? So it sends the synaptic or this the vesicles to the end of the terminal button, and at the very end, there's little, you can kind of see them there, little kind of like ports where the messengers are shot out, and ports where they are received. Okay, so the vesicle fuses with the membrane at the end of the presynaptic neuron, right? The presynaptic terminal end, and that then causes the neurotransmitters that it is containing to kind of explode out of the end and hopefully land into certain marked areas for the neurotransmitters. Okay, they kind of diffuse across the gap, right? It's filled with cerebrospinal fluid, and land hopefully into, again, these little kind of notches. I mean, they don't, not really notches, but kind of look like notches into the postsynaptic neuron. Okay, so what we need to talk about here is that there's these three processes, right? There's the storage of neurotransmitters in the synaptic vesicles. Okay, there's the release of the neurotransmitter molecules into the synaptic cleft, and there's the binding of neurotransmitters at the receptor sites in the postsynaptic neuron. There are three things that need to go right for this to work, right? Each one of these things has to work appropriately, otherwise information is not going to be shared. Okay, when the neurotransmitter and a receptor molecule combine, reactions in the cell cause what's called a postsynaptic potential, which is a voltage change at the receptor site on these postsynaptic cell membranes. It basically causes a chemical change in the postsynaptic neuron. Okay, after producing these, some neurotransmitters, right, they either land and work fine and cause the next neuron to either fire or not fire, depending on what type of neurotransmitter that is. So either they are received or they're not. Okay, so some just kind of get deactivated, right? They kind of get broken down in the synaptic cleft, right? They just kind of fizzle out because there's an enzyme in there that's breaking down the chemical messengers, right? Or sometimes they just literally like bounce out of the synapse. They're not connected, right? There's a gap there. So sometimes those chemical messengers just drift off into the cerebrospinal fluid, never to be found again. So there are those two things that can happen to these neurotransmitters once they're expelled from the presynaptic neuron, right? They can create, again, the postsynaptic potential, which then causes the binding. That's what happens when the binding of a neurotransmitter basically successfully occurs. But there's also another thing that sometimes happens. So neurons don't want to waste chemicals. If there's a bunch of chemicals just sitting around in the synapse that aren't being used, right? They're not landing in a receptor site, and they're not being broken down by enzymes, and they're not floating away. There's something that's called reuptake, okay? And that's when the firing neuron, right? Again, for using kind of colloquial language, the firing neuron basically sucks back up any of those neurotransmitters and recycles those chemicals for the next time that they're needed, okay? The body doesn't like to waste things, so there's something called reuptake, where those messengers are brought back into the neuron. So another way to kind of say it is, I like to think of neurons as really having two functions at the terminals. They either puke or they suck, right? They puke out all these neurotransmitters, and then whatever's left over, they suck back up. They're kind of like my dog when he sometimes eats his own vomit, right? It's this weird, disgusting thing that happens, but that's what's happening with the neurons, and that's information you didn't need to know about me or Murphy or Ralph. Okay, so let's keep going with these how neurotransmission works. Okay, so again, it's similar animation, but it's a little different. So the synaptic vesicles make it to the end of the terminal ends. They then are released into the synaptic gap, right? This shows the synaptic gap a little bit easier. And they either land on receptor sites. These are specific receptor sites that are exactly matched to the shape of the neurotransmitter. That's important for something later, okay? So keep that in your notes, that the receptor sites have a specific shape to them that corresponds with specific neurotransmitters. We'll go over this in class a little bit, but that's an important thing to talk about here. Okay, so if that happens, there's two things that can really happen with these neurotransmitters. There's something called excitatory neurotransmitters. So some receptors act to depolarize the nearby cell membrane. So again, you've got a polarization process happening here. So these neurotransmitters get open up what are called ion channels, and they bring in some of the other, either positively or negatively charged ions into the cell, which then causes the cell to be more likely to fire, right? And increases the probability that the receiving cell will create an action potential or will depolarize. Okay, this is called excitatory. It excites the next neuron to fire, right? You can think of it that way. In other words, this excitatory transmission is basically continue this message, right? Send this, whatever we're doing, keep doing it, right? Pass the message along. It's whisper down the lane without any mistakes, right? It's the game of telephone. It's keep going. This is what we need to keep the information moving, okay? That's an excitatory neurotransmission. There's also, if you can imagine it, something called an inhibitory neurotransmission. So other receptors act to block the depolarization of the receiving cell. So neurotransmitter comes in from one cell and it's basically telling the other cell to calm down, stop doing what you're doing. We want this message to stop, right? This could be if something's overactive, right? A neurotransmitter could come in and say, stop what you're doing. So there are two types of neurotransmission. There's excitatory, which makes it more likely that the receiving neuron will fire. And there's inhibitory, which makes it less likely that the receiving neuron will fire. Okay? So those are the two things. Now, this is where it gets, if this isn't tricky enough, this is where it gets a little tricky. It is not specific to the neurotransmitters. So those chemical messengers in some parts of the body are excitatory, and in some parts of the brain are excitatory, but in other parts of the brain, they're inhibitory, right? So you can't really say like, oh, this is mostly, actually we can say mostly these are excitatory or inhibitory neurotransmitters, but it's not always the case, okay? It's not going to be always the case. So basically what happens is, what I was talking about earlier, you have neurons essentially deciding whether or not to generate an action potential. So I'm a neuron, I'm receiving all this information from a couple of my nearby neurons that I'm getting info from are saying fire, right? They're passing along an excitatory message. A couple other ones that are nearby are passing along an inhibitory message. And as the neuron that's receiving these messages, I'm basically keeping track. And if the excitatory outweigh the inhibitory at a certain level, boom, I release an action potential and I fire my message down the axon to the neurons that I'm connected to. It is this kind of interconnection that creates what we call neural networks and creates really our entire mental world. So there's a lot there. I think it makes sense for us to stop here before we get into specific neurotransmitters and how they work, which we will do in the next video. This video that we just went through, how all this stuff functions, is extremely important for you to know as we move on to these other things. So if you need to watch this a few times, please, I'm encouraging you to do that. We'll also kind of look at my drawings in class, I'm going to draw live for you guys, to go over this stuff as well, because I think it's that important that you hear this, multiple times. Okay, next video, we will be talking about specific neurotransmitters and their functions, or their primary functions. This video was all about how neurotransmission works and how the function of neurons. I will see you guys next time.